Thank you for listening to my presentation. My name is Matt, and today I'll be showing you the wonders of phloem-based long-distance RNAs. The phloem system contains sap, which is used to transport soluble sugars, hormones, proteins, nutrients, and amino acids. And in 1999, long-distance RNA was added to this list. Now, measuring phloem sap isn't as simple as tapping the vein of an animal. Plants of all sizes frequently combat invaders, which may enter through or damage the precious vascular tissue. Woody xylem tissues have many tendons to deter predation. Phloem tissues do not. When damaged, sieve tubes will form a callus, effectively sealing off the section of phloem in question. This process would help prevent the loss of sap, pressure, and help keep out invaders. To tap this sap, scientists have turned to biological and chemical methods. The most destructive method to access phloem sap is to cut the stem, dispose of the first exudate, and sample the next drops, although this may add contaminants from the tissues of severed cells. The use of chelating agents can prevent the formation of a callus, but the use of dissected insect styli, which have penetrated the phloem already, allow for a clean and simple collection. Insects like aphids introduce proteins that prevent callus formation when their style is inserted. The biggest problem facing this method is the slow collection rate. When studying RNA in the context of long-distance transport, it's important to know what kind of RNA is present. In general, there are no ribosomes in the sieve tube, so mRNA does not transcribe any proteins within it. The mRNA may code for proteins in other parts of the plant, however, as was found in gibberellic acid insensitive mRNA in apple trees. MicroRNAs are another form of long-distance RNA. These non-coding RNAs can meet with complementary mRNAs and prevent them from transcribing. miRNAs are known to target regulatory, developmental, and stress response transcription factors. It's suggested that some non-signaling phloem-based miRNAs may be present to prevent the phloem transport of mRNAs, which would wreak mayhem in other areas of the plant. The bulk of research in this field has focused on cultivated herbaceous plants. However, fruiting trees have been given some special attention in recent years. This large concentration on vining and other herbaceous plants should be considered when weighing similarities with the tree field. Is it fair to compare these morphologically different systems? Lending support for the comparison between soft cultivars and trees, Kane Hira and others discovered the first evidence of long-distance RNA signaling in apple trees by searching for mRNA homologs from studies on melons. Using this as the foundation for transgenic experiments in the field can be given additional support, as it is currently commonplace to perform in vivo studies on phloem mobile RNAs from trees in herbaceous plants. The connections only expand past this point. Trees are slow growing and long living. Herbaceous plants grow much quicker and are more conducive to lab growth. RNA coding genes from trees can be marked and inserted into the genome of herbaceous plants. The problem is that common transgenic techniques cause plants to express the inserted DNA across the entire genome. To counteract this, transgenic experiments can be combined with grafting. Rootstocks refer to the mass of the plant that contributes the roots, while scion consists of the shoot or non-root mass. By using a transgenic rootstock and wild-type scions, RNA has been seen to cross grafts and enter the scion tissue against bulk flow. Grafting can be used to attach similar species with dissimilar genotypes. Gibberellic acid insensitive mRNA, or GAI, has been seen to trigger dwarfism in plants, a preferred trait for orchardists. This mRNA was identified in apple trees after it was seen to influence leaflet morphology in tomato plants after transport, and may help to optimize leaf shape and in the current environment. The discovery of GAI mRNA in apple trees triggered the discovery of it in pear trees as well, especially in species commonly used as rootstock. This pear GAI mRNA was seen to travel through the phloem grafts and in a bidirectional manner. Knowing that these mobile mRNAs can travel through grafts, the next step was to assess the consequences of such a thing occurring. As it turns out, drought resistance can be conveyed to a pear scion. Coded for in the rootstock, PBDRM mRNA was found to signal the wild-type scion, conferring a drought resistance, and when it was knocked out of the rootstock, the plant remained drought sensitive. Another pear RNA, PBWOX-T1, when transgenically introduced in tobacco rootstock, altered the wild-type scion's flower morphology. This seems to support a growing idea in the field that long-distance phloem RNAs can influence growth and development. Similar situations may be known colloquially. People have grafted cultivars for desired traits for generations. These mRNAs may not travel through the phloem alone. Early study on phloem-based mRNAs in melons identified several proteins which readily bound to them and were observed to travel with it, through the grafts and within phloem sap. In pear trees, protein PB-WOX-T1 mRNA complexes may form through recognition of UTR domain and long 3' cysteine uracil domains. We now switch our perspective to microRNAs. miRNA 399 is conserved across pumpkins and rapeseed with high concentrations within the phloem of phosphorus-starved plants. It travels from shoot tissues to the roots as a regulator of phosphorus homeostasis, acting to increase phosphorus intake in circulation. Transgenic tobaccos expressing miRNA 399 provide a bigger picture, as it was measured that taking shoots lacking phosphorus would send this long-distance signal to increase root intake. This occurred even if phosphorus levels in the root system were adequate. 11 other long-distance miRNAs are found in common for both herbaceous plants and apple trees. No current studies discuss the transport of miRNA through protein complexes, which may make sense due to their function. Unlike mRNAs, miRNAs may mark their possible transporters for cleavage along with themselves, preventing function in the tissue that would be transported to. The phloem-based long-distance transport of RNAs is incredibly exciting, and recent studies on fruiting trees have started to give woody plants the recognition they deserve. It seems fair to draw parallels between herbaceous plants and trees. However, I believe there should be further consensus around how the phenotypic change of transgenic plants can be compared to their gene-holding counterparts. This field has the opportunity to contribute to food security with a changing and warming climate. RNA signaling over long distances through the phloem has been seen to confer advantages for graft pairs in temperature-stressed regions, changes to nutrient uptake and scarcity, and introduce beneficial phenotype traits such as dwarfism. Thank you for listening.